is where you're going to be interrogated today. Come on in. Just wait right here. I'm Detective Gabe. Good to meet you. Never seen you around these parts, so you need to tell me who you are, and what stands out about you.
I'm Chaetognatha, and I'm considered a deuterostome. My breeding and physical characteristics set me apart from other organisms. My breeding characteristic is idioparous, and I am also ectothermic, which requires me to use heat to regulate my body temperature.
Okay, thank you for being cooperative. That's a lot of detail. Now I need you to tell me where you come from, where you live, and others similar to you.
The Chaetognatha phylum contains two orders, Phremophora and Aphrimophora. I come from marine and estuarine environments. Around a fifth of me and my fellow phylum are benthic, meaning we live just above the deep ocean floor.
Alright, so if I gave you a chance to run away, what mechanisms would allow you to leave?
In order to leave, I would use a locomotion method called balancing. I move through the water column with the help of my tail fins and muscular body. Many of my other phylum friends undergo daily vertical migrations. We rise to the surface at night to follow prey, and during the day we sink, which provides us with protection against predators.
Okay, thank you. What do you want with us humans?
So, on a positive note, I serve as a prey item for a variety of fish eaten by humans. On a not so good note, I may negatively impact humans by passing along parasites to fish that you guys eat.
Okay. Alright, so there's going to be one more question before I lock you back up. Alright? Who's it? Can I help you? So, what are you doing here?
I'm here to bail him out.
Not really how this works, but you know what? I'm going to ask you questions anyway. So, are you the same thing as the other thing? How would you classify yourself?
Yes, I'm also Chaetognatha. We are classified in many ways, including mollusks, arthropods, and nematodes.
Okay, so there's a lot more of you then, is what you're saying. So if there's a lot of you, can you tell me about your methods of reproduction?
Yeah, so we reproduce sexually, and we have separate sexes with males and females, and we each produce different gametes for sexual reproduction.
Okay, now for fuel, in order to stay alive, of course, what is it that you guys eat?
We are predators of copepods, larval fish, and crustaceans.
Okay, you've been around for how long?
Well, we originated during the Cambrian period, around 500 million years ago, when we evolved from protozoans. Here's our 16S tree based on the rRNA sequences. Our taxon, Chaetognatha, is most closely related with these 5 phyla: Chordata, Echinodermata, Annelida, Arthropoda, and Mollusca. This is because Chaetognatha shares a node with them more recently, right here, than it does with the other 2 phyla in the tree. Alright, now moving on to our data matrix tree. This is the tree that was based on the data matrix we developed in class from the dissections of all different phyla. Chaetognatha is most closely related to Annelida because they share a node right here most recently. Now we are going to compare and contrast the 16S tree based on the rRNA sequences and the data matrix tree we developed in class. The trees have quite significant differences between the 16S and the data matrix tree. The data matrix tree is based off of physical characteristics, while the 16S tree is based off of genetic sequences. Both trees have been developed in their most parsimonious forms, but are based on different characteristics. Here's our tree from literature. As you can see, highlighted in yellow are phyla that we are familiar with, such as Echinodermata, Annelida, and Mollusca. Highlighted in pink is our taxon, Chaetognatha. The trees we generated from our observations in lab are different from this tree because this literature tree is based on DNA sequences, while our trees from lab are based on rRNA and physical characteristics. According to this tree, Chaetognatha is most closely related to Annelida and Mollusca because they share the most recent node here. Although we cannot be 100% sure, the tree from literature may be more accurate than the data matrix tree because we had a limited pool of organisms and we were only going off physical characteristics in lab.